Biochemistry? Yes. All right. A lot of ATP today, is that what you said? Or it could be that coffee or whatever you claim is coffee in that cup there, right? Okay. All right. So last time we talked about gluconeogenesis, and I hope you began to see how that relationship is between a catabolic pathway, that is glycolysis, and an anabolic pathway, which was gluconeogenesis, that is breakdown versus synthesis. And you saw that they had what I called reciprocal regulation, meaning things that turned one on, turned the other one off, and then vice versa. Okay? So the Cori cycle, as I noted, was a cycle that takes place in the body that looks like a feudal cycle that I talked about, except for it's not occurring in the same cell. It's only feudal if it's occurring in the same cell. The liver is a specialized organ that's designed to put glucose, among other things, out into our bloodstream. We'll see some other things later that it puts out into the bloodstream, and help the body maintain a certain level. And it turns out that maintaining a certain level of glucose is very important in the body. As I've alluded to the fact earlier, glucose is a poison, meaning that in high concentrations, glucose kills cells. All right? So your body spends a fair amount of time and effort keeping glucose concentration from getting too high. Now, it's an odd poison in the sense that the body needs glucose as well. Your brain relies on the rest of your body to supply glucose. Your eyes rely on the rest of the body to supply glucose because, as I noted, the only tissues that make glucose are the liver, a little portion of the kidney, and the germ cells. Okay? So that means, therefore, that the brain is dependent on a supply of glucose coming in the bloodstream. If you were eating constantly, and yes, I know some people do eat almost constantly, if you were eating almost constantly, then this issue of needing to make glucose wouldn't be there. Okay? But because we're not eating constantly, we have periods of time where our blood supply of glucose is high because it's in our diet, and times when it's low between meals. So when it's low, the liver has to make up that deficit. And what the liver tries to do is it tries to maintain that. Okay? Well, the body really maintains levels of glucose in a couple of ways. One is by synthesis in the liver, and the other is with the hormone insulin. Insulin is a hormone that causes cells to take up glucose. It causes cells to take up glucose. And that has the effect of reducing the blood glucose concentration. That's where glucose concentration is a problem. So we all know that diabetics have problems with high blood glucose levels, in some cases low blood glucose levels, because their insulin is not being either synthesized properly or because their cells have become resistant to insulin. All right? So both of those cause problems because glucose can really get really wildly out of range, and people who have diabetes over time can have very severe problems relating to vision. They can lose their vision. They can lose limbs. Amputation is not an uncommon thing that happens with diabetes. And again, reinforcing that idea that glucose is a poison. Now you're thinking, well, it's a poison in a literal sense, but it's not really a poison in a real sense, to which I say, well, why do you suppose that jellies exist? Jellies exist because they're a way of preserving food, and the preservative is glucose or sugar, okay? Sucrose or glucose, all right? That, you take jelly and you leave it out on the counter for a period of time, not much will happen to it because that high concentration of sugar/glucose that's in there kills the bacteria. You let it go long enough, yes, mold will come and do its thing, but you will not see bacteria that will come and take over jelly. So glucose really is a poison. Okay. Well, the thing I want to move to now, actually I want to just sort of reiterate. My thing is off here. Oh. The thing I want to reiterate at this point is the control mechanism. So you've seen I've talked about control, and there are really three control mechanisms that exist to help cells regulate glycolysis and gluconeogenesis. All right? Now, these are pretty important to regulate because, again, you don't want to break down too much sugar, and you don't want to produce too much sugar in the bloodstream. So the body's got to balance these, and regulation, therefore, is very important. There are 3 mechanisms for controlling enzymes that cells use to do this. One is allosteric, and my battery seems to be dead. Don't tell me. Well, I guess it's dead. Okay, I'll point. There you go. The old-fashioned way, right? All right. You see right there. Anyway, one of the mechanisms is, of course, allosteric, meaning that a small molecule can bind to an enzyme and affect it. So we saw, for example, F26BP was a reciprocal regulator. It turned on PFK at the same time as it turned off fructose 1,6-bisphosphatase. Okay? I didn't really talk about it, but I did mention it briefly, and that is the fact that pyruvate kinase is regulated by covalent modification. Okay? Now, I'm going to say more about that when I talk about glycogen metabolism today, because covalent modification is an important control mechanism for controlling glycogen metabolism. Covalent modification means altering the chemistry of an enzyme. It usually, not always, but usually involves addition or removal of a phosphate group. And we'll see how this affects glycogen metabolism today. The third mechanism is genetic. And what in the heck is a genetic mechanism? Well, a genetic mechanism is control of synthesis of an enzyme. So you will remember that I said that not all cells had glucose 6-phosphatase, the last enzyme of gluconeogenesis. Not all cells had that, so not all cells could produce glucose. Well, if they don't have it, how is it that they don't have it? Their tissues don't express it. So we've already seen, for example, with enhancers, how we have tissue-specific synthesis of certain proteins. Most of our tissues don't make glucose 6-phosphatase. Therefore, most of our cells cannot make glucose. Okay, so those 3 mechanisms help to control the enzymes in glycolysis and gluconeogenesis. Okay. Oh, by the way, another thing I should mention, I'll just mention, you don't have to know this, but the other enzyme, PEPCK, is another one that is not made in all cells. So we see one at the very beginning of gluconeogenesis and one at the very end. So by controlling the synthesis of those 2 enzymes, the body really can control whether or not cells are making glucose or not. So, okay. One pathway that I want to talk about, and all I'm going to do is show you a schematic because you're not going to like it, okay? And I'm not going to make you learn it, okay? We will talk about a pathway related to this pathway later. This pathway is called the pentose phosphate pathway. It's actually related to a pathway in the photosynthesis called the Calvin cycle, all right? I show you this pathway not to get you to learn, memorize steps, or any of that, because it actually is a little complicated in how it does things, but rather to make you aware of the pathway and, more importantly, the importance of the pathway. Okay? So why is this pathway, why do I even bother to talk about this pathway? I talk about this pathway because it provides several very important things for cells. So before I tell you what those are, I want to tell you briefly sort of an overview of what's going on here. Okay? This is a pathway that doesn't really have a beginning or an end. Okay? That already starts, and see frowns already. That already starts it off on the wrong foot. It doesn't have a beginning or an end. Okay? What does that mean? Well, it means that if we look at some of the things that are here, here's glucose 6-phosphate. You've seen glucose 6-phosphate in glycolysis and gluconeogenesis, right? So glucose 6-phosphate can be one of the entry points for this pathway, meaning something can come from those other 2 pathways and feed this pathway. You say, oh, well, that's the beginning. Well, not necessarily. We could look at other things in here and say they could feed the pathway as well. Here's glyceraldehyde 3-phosphate. You've seen that before. Here's fructose 6-phosphate. Okay? Here's another glyceraldehyde 3-phosphate. Here's ribose 5-phosphate. Ribose is a sugar that's in nucleotide metabolism. So any of these could be entry points for sugars from other pathways. All right? Well, what does that mean? Well, what it means is that if we look at this, we see entry points. What are exit points? Well, exit points are anything else that's in there. So I could have an entry point, for example, of fructose 6-phosphate, and I could be putting out ribose 5-phosphate. So I could start with fructose and produce ribose. Well, that may turn out to be very important for a cell that's getting ready to divide, because a cell needs nucleotides, and this pathway provides ribose that's needed for nucleotides. Okay? So this is a very flexible pathway. It's a pathway that takes many different things in and can put many different things out. All right? All right, so what with that? The main things that happen with this pathway, it's a source of two very important things. And that's if there's one thing you want to take away today, I want you to know what these two things are that this pathway produces. All right? One of the most important things that this pathway produces is a molecule called NADPH. Now, we've talked about NAD and NADH, but we haven't talked about NADP or NADPH. All right? NADPH, as we will see when we talk about the synthesis of fatty acids, is used in many anabolic pathways. Not all, but many anabolic pathways. We didn't see it in gluconeogenesis because the cell simply reversed a reaction that used NADH. But when we talk about fatty acid synthesis, for example, they use electrons from NADPH. So this pathway is a source of electrons in the form of NADPH that are used primarily in anabolic pathways. The second thing that this pathway is essential for is the production of ribose. So I've already mentioned that right here. Ribose 5-phosphate is used by cells to make nucleotides. So this pathway provides both of those. And depending upon, again, the needs of the body and the cell, all right, then this pathway will adjust to making more NADPH or making more ribose 5-phosphate, all right, and dealing with many other things that are there. So that's basically what's up with the pathway, all right? Okay, as I said, we'll talk about the Calvin cycle later in the term, because the Calvin cycle is, in fact, related to this pathway, and I'll talk just a little bit more in more detail about that when that comes up. Okay. One of the things that this pathway does that I also, actually, no, I'll save that. I'll save that. Let's not do that. All right. So let's turn our attention then to glycogen metabolism. All right. So glycogen is, I will remind you, a polysaccharide. And it's a polymer of glucose. It's the primary storage form of glucose for animals, including us. Okay? And it is long chain. It consists of long chains of glucoses in alpha-1,4 linkages with branches of alpha-1,6 about every 10 to 20 glucoses. So here's a structural schematic of glycogen. And we can see that glycogen has branches. Again, on this one, it's about every 10 or so residues, and you can see them all branching off. Glycogen is therefore a very, very branched structure. Glycogen is stored primarily in your liver and in your muscles. Primarily in your liver and in your muscles. The muscles use it for a very immediate source of energy. A very immediate source of energy. They can break down glycogen pretty quickly when a signal comes and give themselves a very good dose of glucose. Now, one of the things that causes cells to break down glycogen is a hormone known as epinephrine. You probably have heard of epinephrine as adrenaline. They're the same thing. Okay? Epinephrine equals adrenaline. Adrenaline stimulates many things, but one of the things that it stimulates is the breakdown of glycogen. Well, the breakdown of glycogen, as we will see, provides a source of glucose. So the reason that you hear these stories about people who see some terrible thing that happened on the highway to a family member or something, and the person goes out and grabs the rear end of a car and picks it up and moves it, those stories actually, in most cases, are true. They're able to do superhuman things because what's happening is their body is producing a tremendous amount of glucose that their muscles are using, they wouldn't otherwise normally produce, and they go and do their thing. Okay. Yeah, it's pretty crazy. It's true. All right? So that, as we will see, is important in the regulation of glycogen, regulation of the metabolism of glycogen. Okay. Well, when we think about glycogen, we've got two things to consider. We've got synthesis and we've got breakdown. So we'll start by talking with about breakdown first. Why do I have that there? Why do I put that? Oh, okay. It does say glycogen. I read that as glucose, but it's glucose N. I thought, whoa, I don't even know my own figure. All right. So glycogen, when the body wants to break down glycogen, okay, it starts with what you see on the screen, a glycogen which has an N number of glucoses that are there. You saw in the previous figure that there were a whole bunch of N's. All right? A whole bunch of N's. Let's make sure we emphasize that point. There's an end. Every blue thing on there is the end of a chain. Okay? So you see a lot of ends of chain. And as I noted before, glycogen is broken down, starting from an end and moving inwards. So the more ends that you have of glycogen, the more glucose can be produced rapidly. Okay? So we've got glycogen, and we've got a bunch of ends. All right? Glycogen does something, I'm sorry, the enzyme that breaks down glycogen does something interesting. First of all, let me give you the name of the enzyme of glycogen. It's called glycogen phosphorylase. P-H-O-S-P-H-O-R-Y-L-A-S-E. Glycogen phosphorylase. Glycogen phosphorylase catalyzes a very interesting reaction. You see it on the screen. Here's glycogen. The enzyme has grabbed a hold of a phosphate and one of those ends. And what it does is it uses the phosphate to break off one of those glucoses. So you see that glycogen is left with N-1. And it puts the phosphate on position number 1. It makes something called glucose 1-phosphate. Okay? Now I'd like you to think back to that first reaction I talked about in glycolysis. When I talked about glycolysis, we started with glucose, and we added ATP and hexokinase, and the phosphate was transferred from ATP onto glucose to make glucose 6-phosphate. And I said that the energy of ATP was needed to make that happen. My question to you here is, why isn't energy needed to make this reaction happen? Because we're putting a phosphate onto glucose. What do you think? Why is ATP not added to put this on? Greater concentration of reactants, that actually will help to drive a reaction. You're correct. But there's another more important reason. What's that? More stable? Not quite. Back here? There's not an oxidation, because again, we don't see NAD, but nice try. Dark magic, that's it. Gold star for the day. All right, it turns out that what this thing is showing you, we don't have exactly the same thing we had before. We had glucose before, we've got glycogen here, right? The enzyme is breaking a bond between glucose and the rest of glycogen. That's an alpha 1,4 linkage. What this is telling us is that there's energy in that linkage. So the energy in breaking that bond is used to put the phosphate onto glucose. Consequently, we don't need ATP to put that phosphate on. So the energy for making this reaction go is found in the bond between the glucose and the rest of the glycogen. That's an alpha 1,4 bond. Okay? Well, why does the cell bother to do that? Why doesn't it just use water to clip off glucose? Well, it turns out the cell is saving energy. Just like putting an LED light bulb in your house will save you energy, so too will putting a phosphate on to glucose without any ATP is going to save you energy, right? If we started with bare glucose and we want to get to glucose 6-phosphate, put a phosphate on there, we've got to use ATP, right? We've got a glucose with a phosphate on it, and it didn't take any ATP. So putting a phosphate on, using the energy of that alpha 1,4 linkage, saved the cell energy. All right? You might say, well, I can't use glucose 1-phosphate in glycolysis. I need to have glucose 6-phosphate, to which I would say, yep. Now we get to the next step. All right? And the next step is this. Glucose 1-phosphate can readily be converted into glucose 6-phosphate. Aha. Now we've got an intermediate, glucose 6-phosphate, that can feed directly into glycolysis. It can also, if this is occurring in the liver, lose that phosphate and become glucose so it can be dumped into the bloodstream, right? So this turns out to be important in two different ways. One is it can directly feed glycolysis if it's a muscle cell that needs the energy. Number two, the phosphate can be clipped off if this is occurring in the liver because glucose 6-phosphatase is present and the glucose can be dumped into the bloodstream. So glycogen can be a source of energy, okay, for cells that need it. Glycogen can be a source of glucose for a liver that contains it. Everybody with me? Okay. Now, turns out there's one, and this, by the way, this is one of the simplest pathways you'll ever learn. All right? We're almost done with it, actually, already. Yes? Okay, so his question is, did it take any energy to store glycogen? It does. And so that'll be coming up shortly. So it's a very good question. And in fact, it does take additional energy to make and store glycogen. Yeah. Or make it, basically. Okay. So we're almost done with the pathway. We've got one other thing we have to consider. And that's this. If we look at the cell, if we look at the glycogen within a cell, glycogen phosphorylase will not completely break down glycogen. And the reason it won't is that glycogen phosphorylase will only cut alpha 1,4 bonds. You remember we have alpha 1,6 branches as well. Okay? It turns out that not only that, but glycogen phosphorylase doesn't even like to get close to a branch. It will only go to about 4 residues away before it stops. It's a very finicky, picky enzyme. It won't get any closer than about 4 glucoses away from a branch. All right? Well, if you're going to break down glycogen completely, it means that you need another enzyme. And the cell has another enzyme that has a long name that's about this long that I like to call debranching enzyme. Debranching enzyme does something kind of cool. It clips off 3 of those 4 glucoses from the branch and transfers them to the other end. So you see these 3 guys right here are being attached right here. To refresh your memory, this bond right here where my arrow is an alpha 1,4. And this bond that's being formed down here is also an alpha 1,4. No additional energy is needed to break a 1,4 and make a 1,4. All right? But then the enzyme does, that's the first thing that the enzyme does. The enzyme actually does 2 things. The second thing that the enzyme does is it's got 1 glucose left right here, so it uses water and breaks it off. It doesn't use phosphate. My question to you now is, why doesn't it use phosphate? Any thoughts? It's using water, and it breaks off that last glucose from the branch. You just said it right there. There's not as much energy in a 1,6 bond. So there's not enough energy in a 1,6 bond to allow the addition of a phosphate to it. Therefore, the cell does the only thing, or the enzyme does the only thing it can do, which is clip it off, and it uses water for that purpose. Very good. Very, very good. Okay. So at this point, we now have a straight chain of glucoses, and glycogen phosphorylase goes, oh boy, and starts chewing in one at a time until it gets within 4 of a branch again. Any nearby branch, that's correct. Okay? That's glycogen breakdown. Now we'll see that there's some considerations about regulation, but in terms of the enzymes, you saw 3 enzymes there, only 2 of which I gave names to. One was glycogen phosphorylase. The other was debranching enzyme, the one that converted glucose 1-phosphate to glucose 6-phosphate. I didn't give a name because I didn't want you to have to memorize it, because if I gave it a name, then you'd have to memorize it. Right? I'm going to give you the name? No. All right, we won't give it a name. All right, so that's how we break down glycogen. Very simple. Now we need to synthesize glycogen. Well, synthesizing glycogen, as he asked right here, requires some additional energy. We can't just reverse the reactions that were used to break down glycogen because we've got to make some bonds that require some energy. Okay? Remember that anabolic pathways tend to take more energy than catabolic pathways? We saw that gluconeogenesis took 6 triphosphates, but glycolysis only gave us 2 triphosphates. Okay? So it takes more to build than it does to break down. Okay? Somebody asked me after class the other day, well, if it takes more to build than to break down, then how do we balance that out? How do we balance it out? And the answer is we don't. That's why we eat. The reason we eat, if we balanced it out, we would never have to eat, except to grow. If we balance it out, we would have perfect balance between synthesis and breakdown, but we don't have that. For the same reason a car, you have to continue to fill up the gas tank. Right? We need to fill up that difference, and that's why we eat, so that we can make up that difference between catabolism and anabolism. All right, so we need to put in some additional energy to make glycogen. All right? So we don't use glucose 1-phosphate as a molecule to add to glycogen. Instead, we use something called UDP-glucose. UDP-glucose. Gesundheit. UDP is a nucleotide. In fact, if you look at the nucleotide, it goes from here, there's the base, there's the sugar of ribose, there's the diphosphate, and that UDP, which is this thing over here on the right, that UDP is attached to a glucose. It turns out this molecule right here has a lot of energy in it. It's something we call an activated intermediate. An activated intermediate is a molecule that has a lot of energy in it. And it uses that energy to donate a part of itself to something else. The something else in this case will be a growing glycogen chain. So I'll repeat that. An activated intermediate is a molecule that has high energy, and it uses that energy to donate a part of itself to something else. Well, where do we get UDP-glucose from? All right. UDP-glucose is made. Oh, I thought I had it on here. I don't have it on here. Blast it. All right. I'll have to tell you where it's made. All right. UDP-glucose is made by starting with the following 2 things. UTP, the nucleotide, and glucose 1-phosphate. You've seen that before. Glucose 1-phosphate plus UTP, and an enzyme whose name is even longer than that, right, gives UDP-glucose. Now you'll notice UTP has high energy because it's a triphosphate. It takes a triphosphate to make this molecule. That's another piece of information that says that this guy has high energy in it. UTP plus glucose 1-phosphate gives this molecule. All right? This molecule is what the enzyme that makes glycogen will use to add glucose. So this part over here on the right is going to be added to a growing glycogen chain. Okay? The enzyme that does this is known as glycogen synthase. S-Y-N-T-H-A-S-E. Glycogen synthase starts with glycogen, adds or takes a UDP-glucose, and transfers the glucose onto a glycogen chain, making an alpha 1,4 bond. Now you know why an alpha 1,4 bond has energy in it, because it took a high energy intermediate to make it. All right? As a consequence of this, the glycogen chain has grown by one more unit, or one more glucose unit. We're almost done. We're almost done. We've forgotten one thing, though. We've got to make branches. Well, again, the enzyme that catalyzes the synthesis of branches has a long mouthful of a name, and we're going to discard that and call it branching enzyme. And what does branching enzyme do? Okay? Now, I could make you guys memorize how far from the end it cuts and exactly where it puts it and so forth, and I think that would just be mental dumbness, right? It wouldn't make any sense to make you memorize it. If you know that branching enzyme grabs a section of a glycogen chain and converts it into an alpha 1,6 linkage, you know what branching enzyme is doing. What's doing the original one? Well, originally, this branch had to have come from branching enzyme. But all these alpha 1,4s, remember these are all alpha 1,4s out here, and all these are alpha 1,4s. Only the branch is 1,6. All the 1,4s were made by glycogen synthase. So this branch had to have originally been made by a branching enzyme. Correct. That's correct. Okay? All right. So branching enzyme does that. You'll notice that no ATP is needed. And based on what I told you, that sort of makes sense, because you knew that alpha 1,4 bonds had more energy than 1,6 bonds. We're converting a 1,4 to a 1,6, meaning it's going to be energetically favorable, because 1,4 bonds have more energy than 1,6s. We're breaking a 1,4 and making a 1,6. It will give off a little bit of heat. Yes? Yep. All right. So we've broken down glycogen. We've made glycogen. I told you they're very simple pathways. Now, being a biochemist, I'm required by law to make it complicated. All right? So I'm going to show you the regulation of this pathway. I don't do that to scare you. All right? I'm being a little silly, actually. But I want to show you the regulation, because the regulation is kind of complicated. But there's some very, very interesting things metabolically about the regulation. I've got some time today, and I'm going to tell you some of those things. Okay? All right. So let's look at the regulation of this pathway. All right? Wow. This is only the regulation of the glycogen phosphorylase. And by the way, that's the only one I'm going to show you the regulation of. I'm not going to show you the regulation. Glycogen phosphorylase is regulated in a very complicated way, and glycogen synthase is also regulated in a complicated way. Okay? But if you understand this, you're doing pretty good. I'm going to take you through it. All right. So what do we see? What do we see is going on here? Well, what this is showing is the glycogen phosphorylase itself. It actually occurs in 4 different forms. 4 different forms. Dr. McFadden talked about R states and T states with respect to proteins. He mentioned it. Okay, well, I'll mention it in a little bit more detail. All right? So proteins that do things, like hemoglobin, like enzymes, all right, can occur in two different states, R and T. R stands for relaxed, and we generally associate R with being more active. T stands for tight, and we generally associate it with being less active. Okay? Hemoglobin, for example, exists in an R state and a T state. In the R state, hemoglobin grabs a hold of oxygen. In the T state, hemoglobin lets go of oxygen. An enzyme that's in the R state will be what we think of as turned on, and an enzyme in the T state we will think of as being turned off. Okay? All right? If we look at this enzyme, we see that one of the forms of the enzyme is in the T. Another form is in the R. This form up here has phosphates on it, and because it's on the top row, it's in the T. In the bottom, it has phosphates, and it's in the R. So the four forms are as follows. No phosphates, T. No phosphates, R. Phosphates, T. And phosphates, R. We're talking about glycogen phosphorylase. All right? Yeah, okay. Don't worry. I'm going to talk you through it. Talk through it. Okay? Let's first talk about the T to R with no phosphates. All right? The T form I've told you is basically an off switch. If I have glycogen phosphorylase present with no phosphates on it, okay, it's called the B form. So these two guys we see on the left are the B form of the enzyme. When the B form of the enzyme is in the T state, has no phosphates, it's not active, it sits there, it does nothing. When it converts into the R form, it becomes active. Well, how does it convert from R to T? One of the things that, if I had taught you about enzymes, I would have told you is that things convert from R to T by allosteric means. Molecules cause that change. Molecules cause that change, or they can cause that change. All right? Here's a cell that has plenty of ATP. And if a cell has plenty of ATP, what would you say about its energy state? High energy. Does a cell that has high energy, let's say it's a muscle cell, does that muscle cell want to be breaking down glucose if it has plenty of energy? Nope, it doesn't. Okay? So, it has ATP. When there's plenty of ATP, ATP binds to glycogen phosphorylase without a phosphate and converts it into the T state, thereby turning it off. No glucose, no glycogen is broken down. Glucose is not needed. For most of you sitting there right now, that's exactly what's happening because you have high ATP because you're not running and jumping and playing. All right? That's what's happening. Glucose 6-phosphate is similarly an indicator of high energy. So either one of these guys can bind and turn this guy off. The fire alarm goes off. We've got to get out of here. We've got to run far away. Our ATP levels quickly fall. Our AMP levels quickly rise. And look what happens. AMP binds and converts this guy from the T state into the R state. And in the R state, it's active, and it starts breaking down glycogen because the cell needs energy. Makes perfect sense, right? Everybody understand that? High energy, the enzyme's turned off. Low energy, the enzyme is turned on. Okay? Any questions about that part? We've just gone up and down on the left side. Let's think about, I'll tell you later how the phosphates get on there and how they get off there, but let's think about the other form that has phosphates on it. Okay? If I'm a liver cell, and I've been dumping a whole bunch of glucose out into the bloodstream, and I was running, but I've stopped running, my muscle cells aren't going to be burning that glucose, right? So my blood glucose levels are going to rise. My liver is going to sense that, hey, the blood glucose levels are high. We're not going to put any more glucose out. So the liver glucose concentration starts getting high. And when the liver glucose concentration gets high, do I want to continue to break down glycogen? Absolutely not. Glucose binds to this form and turns it off. Okay? So when glucose levels get high, the phosphorylated form gets turned off. Now you'll notice that there's an arrow on the right side and there's nothing on it. That means that if there's no glucose present in this cell, that the enzyme is automatically going to flip from the T to the R. It doesn't require, okay, it doesn't require anything to convert from T to R. It's for this reason that people say that this form of the enzyme is more active, because it's more common that there will be no glucose than it is that there will be glucose. So if there's no glucose, this guy is flipping from top down to bottom. It goes from T to R. Everybody's happy. Okay? Now what I've just described to you are the allosteric regulation of these two different forms of the enzyme. I haven't told you how I go from B to A. I'm going to tell you that in a minute. But I've told you how these are regulated. ATP, G6P will turn the unphosphorylated form off. AMP will turn the unphosphorylated form on. Glucose will turn the phosphorylated form off. And if there's no glucose, the phosphorylated form will be turned on. Well, now we look at this and we think, well, what is it that converts the phosphorylated and the dephosphorylated forms into each other? All right? Remember I told you about epinephrine and adrenaline? Adrenaline is a hormone. And this hormone binds to target cells, in this case, muscle or liver cells. And when it binds to that hormone, it stimulates the addition of phosphates to enzymes. Adrenaline / epinephrine stimulates the phosphorylation of enzymes, meaning the addition of phosphate groups. That's a covalent modification that I talked about earlier. Adrenaline stimulates that. Look what adrenaline's doing, guys. It's converting dephosphorylated forms into phosphorylated forms. And the phosphorylated forms are much more likely to be activated. If I'm out there and I see my child with a car sitting on top of part of it, right? Panic, fear, immediately I burn glucose, immediately I make adrenaline, immediately glycogen, phosphorylase becomes phosphorylated, and immediately it starts going into the R form and breaking down glycogen, and breaking down a ton of it. Bang, 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 just like that. This can happen in seconds. It doesn't take very long for your adrenal glands to release epinephrine. It doesn't take very long. It doesn't take hardly any time at all for the cells to respond. That's cool. Now, if we left the phosphate on all the time, we'd have a problem, because we would very rapidly break down all of our glycogen. We don't want to leave this in the on state forever. We want to turn it off somehow. Okay? Well, we turn it off in an interesting way. All right? Let's imagine that I've just eaten a meal. I'm not exercising. I'm not doing anything. What happens to my blood glucose levels? High. And what does my body release when my blood glucose levels get high? Insulin. And guess what? Insulin stimulates this enzyme that takes phosphates off of things. Adrenaline favors the breakdown of glycogen. Insulin stops the breakdown of glycogen. It's even better than that. All right? I'm not going to show you, but I'm going to tell you. Glycogen synthase is reciprocally regulated from this guy, at least as far as phosphates go. The same enzymes that put phosphates onto this guy put phosphates onto glycogen synthase. When adrenaline comes into the cell, or adrenaline stimulates the cell, it phosphorylates, the cell phosphorylates glycogen phosphorylase. It also phosphorylates glycogen synthase, but because we have reciprocal regulation, phosphorylation of glycogen synthase turns it off. We turn on the breakdown enzyme at the same time as we turn off the synthesis enzyme. We don't want them both going on at the same time, or we've got problems. Dephosphorylation does the opposite. When insulin comes along, it stimulates the removal of the phosphate from glycogen phosphorylase and the removal from glycogen synthase. That has the effect of turning on the synthase and turning off the phosphorylase. The last thing I'll say before we do a song, the last thing I'll say is that glucose is a poison. Insulin is stimulating the cell to take up glucose, and it's stimulating the cell to take up a poison. Isn't the cell going to be in trouble? The answer is no, because glucose is being converted into glycogen. We're turning on the synthase. So that glucose comes in, it doesn't stay there and poison the cell. It becomes glycogen, and glycogen is not a poison. Okay, that's a lot of stuff. Questions before I turn to anything else? I was going to tell you about, the first thing I'm going to tell you about tomorrow is how caffeine works. Because caffeine plays into this, and it's very cool. Yes? Okay. So this enzyme, glycogen phosphorylase, is active, all right, when AMP is present. Has nothing to do with glucose. Yeah. Okay? It's the presence of energy state and or the phosphorylation of the enzyme. Because the phosphorylation of the enzyme is going to favor this, and then that automatically will turn on to this. Okay? Okay. I have a song that's a structural song for you. Let me pull that up for you. It's about the structure of sugars. It's to the tune of Brahms' Lullaby. All right.